She's going to be talking about her research. Sorry, there's sirens going by. So I guess, Alvin, I'll just ask you a few questions about how you got to where you are before you start. So did you always want to be a scientist growing up?
Yeah, that's a great question. So I grew up in Florida, so I was always outdoors on the beach or running around in my backyard. And I really enjoyed being in nature. And so that kind of spurred me into thinking about science. But I didn't know I wanted to be a scientist until I was in high school. And that's when I really realized I enjoyed asking questions that we don't really know the answer to. And that's a really fun part of science, is you get to explore something that no one else really knows the answer to. And that's why I really enjoy being in science.
Cool. So I guess going hand in hand with that, what do you think is one of the harder things about being a researcher and a scientist?
Yeah, being a researcher or scientist, it can be frustrating at times because you don't know the answer. And so you may think you have an idea of how something works, and you may go down that route of doing experiments. And then you only realize later that actually you probably should have done something else, because that really isn't going to answer the question you wanted. So sometimes that can be a little frustrating because you work really hard to try and address the question, but then maybe it doesn't quite work out the way you think. And so it can be a little bit hard, sometimes a little bit frustrating if you don't really understand how to address something.
I think it's interesting. Like, we've had so many people in so many different fields give presentations, and that's like the same answer that we all have. It's like, that's the most frustrating part, which says something about, like, you know, the struggles that we all face. But then I guess opposite of that is, what do you think is the most rewarding part?
Oh, I just love sort of the community of being a part of a group of people who are really excited to explore new topics across different fields, and being able to communicate that to other people to help out society. I think the people behind science is sort of the best part and the most enjoyable. Not only like the people within science, but also communicating and helping others. That's what I love about science.
Awesome. Cool. Well, thank you. I guess we'll let you get into it. That was good to hear.
Sounds good. And feel free, if you guys have questions, you can ask them in the chat, and Aubriana and Natalie are happy to ask them, or you can unmute yourself if you feel comfortable with that. There'll be sometimes in the slides I'll ask you a question, and you can feel free to message in the chat or unmute yourself.
Yeah, we'll keep an eye on it. Cool. Okay, I'll mute myself and let you take it away.
Sounds great. All right. So my talk today is titled Mitochondria on the Move. So I'm a second-year graduate student at Columbia University, and I study mitochondria. So a little bit before we get into it, I'll give you a little bit of background. So I come from a chemistry background, and so chemistry is the study of molecules. And I really love research, like I said, because I love being in the lab and asking questions that we don't know the answer to. But science is a lot of fun. So being able to communicate what we learn to other people and to also hang out with other scientists can be a lot of fun, and creating a community of people who really care about learning new things. But outside of science, I have a lot of other hobbies. I enjoy playing the clarinet. This is sort of something I've done on and off since I was in middle school. And I think it's just a really relaxing and fun thing to do. I also really like being outside. This was a picture I took a few weeks ago in Central Park. There's a little Dalmatian hanging out of a fire truck, and I think it's really cute. But I try and balance being in the lab with being outside and having other hobbies. And I think this is a really important part of being a scientist, is also having other fun things that I get to do. So about my research, I study mitochondria. So mitochondria are an organelle. And in this sort of picture, we have mitochondria in orange. And you can see that as an organelle, mitochondria are a small part of a cell. So they have a very specific function within the cell. And they play this role to help maintain the sort of healthiness of the cell and the function. So you may think of other organelles like the nucleus. So the nucleus is where all of the cell's DNA is stored. So the DNA is all the instructions that the cell uses to encode all of its important little proteins and things that it needs to stay healthy. So mitochondria is just one specific organelle. And mitochondria have a really interesting history. They actually used to be bacteria. And so here we have a cell shown in this star-shaped yellow. And in this cell, we have the mitochondria in green actually being engulfed by the cell. So millions of years ago, mitochondria existed as independent bacteria. And so as independent bacteria, they didn't rely on the cell at all until they became engulfed. And once they became engulfed, the cell started to depend upon those engulfed little bacteria, and they became mitochondria. So over time, the mitochondria depended on the cell, and the cell depended on the mitochondria. And this is a really interesting phenomenon, and it's also known as symbiosis. So symbiosis is when there's a relationship between two independent things, and usually it's considered a positive thing where they benefit each other. And so in the mitochondria and in the cell, the mitochondria benefits because the cell provides the protection and also provides the nutrients the mitochondria needs. And the cell benefits because the mitochondria is able to provide nutrients for it. And so this symbiosis is sort of like in Finding Nemo when we have the clownfish and the sea anemone. So the sea anemone provides the protection for the clownfish, and the clownfish provides waste, which is nutrients to the sea anemone. And so this is mutually beneficial, and it allows the sea anemone and the clownfish to exist and have a really great relationship. And this is just like mitochondria in the cell. So what do mitochondria do within a cell? They make energy. And so energy is really important because this is what the cell uses to do all of its important functions. And so energy within the cell is called ATP, or adenosine triphosphate. And adenosine triphosphate, or ATP, you can think of like a currency. And so whenever the cell needs to do something, it uses ATP in order to do it. And so how is this energy made? Because if this energy is so important and it's used in so many different things, where does this energy come from? And so energy comes from our food that we eat through glucose. And so glucose is found in things like honey and sweets and carbohydrates, which are like starches. And so when we eat food, this glucose comes into our cells and it goes into our mitochondria. And that, in combined with oxygen, so that's from the air that we breathe. So the food we eat and the air we breathe helps our mitochondria make ATP, or energy. And so this energy is isolated in the mitochondria, which is in this specific organelle within the cell. But the ATP is actually able to go to all over the cell. So that way it can do all the different functions. So how do cells use this energy or this ATP? So they do all sorts of things. They do recycling. So using different proteins and breaking them down, building them into new things. Or they use them to maintain ions. So an ion gradient is a gradient where we have all sorts of chemicals and we have them pushed against their gradient. So normally ions want to flow from an area of high concentration to an area of low concentration. But sometimes the cell needs it to go the opposite. So we need to push ions out of that area. And in order to do that, we need energy, or ATP. And so all of these different roles and many more that the cell uses requires the energy, ATP, that is made by mitochondria. So let's think about this. What parts of the body do we think need lots of energy? So we have things like our hair, we have our heart, we have our head and our brains, our fingers and toes. What do you think are the parts of the body that need lots of energy? Feel free to type in the chat or to unmute yourself and think about what parts of the body need the most energy. All right, the liver. That's a really great guess. The liver, the muscles. Yeah, the muscles. Any other guesses? The brain. Yes, you guys are doing fantastic. So all of these and more. I didn't actually put liver on the slide, but liver definitely needs a lot of energy. And so the heart, this is an organ that pumps blood throughout the body and it oxygenates our blood. And that is an organ that never gets tired, right? So even when we're asleep, our heart is constantly pumping. And so it definitely needs energy all the time. And then we have our brain, and our brain is, again, always doing different functions. We have cells within our brain communicating with each other, and that's going on even when we're asleep. And then we have our muscles, and our muscles are needed for doing all sorts of activities, whether we're playing outside, kicking a ball, or we're getting up from a chair, or we're moving around. And so all of these parts of the body actually really have a high concentration of mitochondria because they require so much energy and they're going all the time, right? We always have our heart pumping and our brain is always thinking. And so all of these parts are really important for mitochondria. I study mitochondria in the brain. And so the brain is a really interesting place to study mitochondria, like the other places, but most importantly, because it's really energy demanding, right? So the brain is a tissue, an organ that has lots of different cells within it. And those cells require a lot of energy. And that's because they do things like recycling. They also maintain ion gradients. So all those different functions that I said earlier that the cells use for energy, the brain uses. And so it's one of the most energy demanding spots in the body next to the heart. Additionally, the brain has a lot of complex processes. And so there's a lot of communication that goes from cell to cell. And so your body is really controlled by your brain. So it tells us what to do, whether it's to speak or to look around, to move our muscles. And so mitochondria in the brain are really important to provide the energy needed for those cells to communicate with one another and to do so in a really fast manner because we need to do actions and to say things quite quickly. And so our brains need to constantly have the energy needed to do those things. Additionally, brains have a lot of cells. So unlike our skin, which regenerates all the time, we're constantly shedding cells. The brain has all of these cells and they stay with us our entire lives. So we're never getting constantly renewing cells in our brain. They're always with us. And so we really need all of these cells to be really healthy for our whole lives. So that way we can continue to have all of our normal functions and we're able to stay healthy. And so that brings us to disease. And so diseases that affect the brain often involve mitochondria. And so if we want to learn more about those diseases, or if we want to find treatments for those diseases, we need to understand how mitochondria function in the brain and how we could actually target them to create treatments for those diseases. And so for all of these reasons, I study mitochondria in the brain. And I think it's a really fantastic place for science because we still don't know quite a lot about mitochondria in the brain. So what types of cells are in the brain? And so there are a lot of different types of cells, but one of the main types of cells in the brain are neurons. So neurons are a particular type of cell that transmit electrical signals and communicate with one another. So here we have a neuron and it has these really long branches. And you can see in this whole network, we have neurons that are communicating with one another and forming this really big mass. So neurons are this really special type of cell, like I said, and there are just so many within your brain. And so they need to communicate with one another and form this really intricate network. And forming this network requires a lot of precise movement. And part of that is given through energy provided by mitochondria. So mitochondria are located throughout these cells. And that's why we think that mitochondria are really, really important not only in development, but also throughout your entire lifetime, because they help these cells maintain this network and communication from cell to cell. Neurons are dynamic. And what that means is that they're constantly changing their inputs and outputs. So there's different parts of a neuron. There's the dendrites, which are shown up here on top. And dendrites are special parts of the cell that receive input signals. And so these input signals propagate from a different cell, and they come through the dendrites, and they go through all the way through the cell down to the axons, which transmit the signal to a different cell. So we have dendrites which receive inputs, and we have axons that transmit those outputs to other cells. So for example, you could see something in your surroundings and your eyes are going to interpret that, and it's going to go to your brain, and your brain is going to have neurons that are going to see some sort of transmission of that change in your visual scene. And then that change is going to go through all different neurons. Perhaps it'll go towards your motor neurons, so it'll get you to move. Maybe it's a really scary thing and you're going to need to run away. So your muscles are going to need to run and get into quick motion. So your neurons are really important because they need to make really quick decisions. They need to transmit information from cell to cell really quickly. And we call them dynamic because these inputs and these outputs can change with time, right? Like you never know what's going to come into your visual field. You can sort of anticipate what might happen, but your brain needs to actually transmit what's happening in your visual scene, for example. And so what we can do is we can actually measure what the neuron is actually seeing, what the input of that neuron actually is. And so if we look at that, we can see this is a signal that is coming through an input to the dendrite, and we can see it as this bright white light that's transmitting or moving along. And what we're seeing here is calcium. This is a calcium wave. And so calcium is a specific signal that neurons use to transmit an electrical change that happens across their dendrite. And here it's really cool because you can actually visualize this by this bright white light that changes in across the entire dendrite. And so here we're actually seeing this movement of calcium from the tips of the dendrite moving down, and the signal is going all the way down, and we're seeing it end here, but it goes all the way down towards the axon. And so all of this input of signal and the propagation of this calcium, this requires a lot of energy. And that's why we have mitochondria drawn here in this green color, distributed throughout the entire neuron, because energy is required not only where there's an input of signal and we see this calcium wave, but also where there's an output of signal. So where we're trying to show a signal going from one cell to the other. So mitochondria have many unique shapes. So far you might have seen them as really small, round, tubular things, but actually when they're in the cell, they really make these really cool shapes. So here we have on the left, mitochondria in this dark shape, and they kind of look like thin spaghetti. They're kind of really long and tubular, and they're kind of interconnected with each other. And you can see here in white is the neuron itself. This is the dendrite. And it forms these really interesting spiny hooks, and it has this really long tubular shape. And the mitochondria kind of goes along with that cell and follows along with its shape, but also kind of melds its own sort of way. And in different parts of the cell, we see there are different shapes of mitochondria. So in the dendrite, it has this long spaghetti-like shape. But in the axons, it has this sort of round, sort of compact structure. And so we can see that in different parts of the cell, mitochondria have, they come out in different shapes. But we don't actually really understand why that is. So one idea may be that in the dendrites, this long spaghetti tubular shape helps that calcium wave, which I showed before, that bright light that moves through the dendrites. Perhaps this long tubular mitochondria helps that signal to propagate through the cell. So perhaps that calcium wave is helped move through the cell by these long tubular mitochondria. But we're not really sure sort of why mitochondria are different in these different parts of the cell, and we don't fully appreciate what the molecular underpinnings are. So what actually tells the mitochondria to be a different shape? That's something we're still investigating, and it'll be really important to try and understand perhaps different functional differences of mitochondria in different parts of the cell. So part of my work has looked at mitochondria shape. And here you can see I've reconstructed mitochondria in 3 dimensions. And you can see I've colored the mitochondria just based on differences in sizes. And so you can see this one teal mitochondria, it's just one mitochondria, and it's huge. It's going from all the way to the bottom, all the way to the top, and it branches. It has sort of a Y shape. It gets bigger in some areas and it gets smaller in others. What you don't see is the neuron here. And so I haven't reconstructed in this image the neuron, but it encompasses this whole area that the mitochondria is part of. This is particular to a region in the dendrite. And so you can see this sort of long tubular shape is similar to like the spaghetti size mitochondria we see over here. But we also see some of these smaller mitochondria, which we found in the axon. And so they're small and they're round, somewhat more tubular. They kind of have a more regular shape than these long tubular ones. So this is going to be, oh, go ahead, Aubriana.
Um, has anyone done like live imaging where they see like a cell, uh, well, like a cell before it reaches out its projections and like see like do the small more round mitochondria that sort of start at the soma at the like the main body, do they stretch out or is it more like mitochondria kind of like helping each
Yeah,
other, like fusing together to make these long ones?
that's a really great question. So that's something that is still being investigated, but we do know that mitochondria can do all sorts of dynamic activities. And so the coming together, so the bringing of fusing of 2 mitochondria is called fusion. And that's something that we actually have seen in vivo. And I haven't shown any images of that, and I don't think I have any today. But that is something we have seen. So where 2 mitochondria, which are maybe smaller, they come together and they form a bigger mitochondrion. And that's a really cool process. We've also seen the opposite, where a big mitochondrion is fused together and then it comes and it goes apart, and we come out and see 2 mitochondrion. And so there are differences in how mitochondria come together and apart. And we kind of understand there's these different molecules and proteins that control when they come together and apart. So they're called kinesins and dyneins, which are different proteins that are used for transport. But what we still don't understand is how that's controlled at the global level, right? Because what we know is that mitochondria need to be at certain places. And so if they're all fragmented or they're all fused into just one bit, then maybe that leaves other areas with either too many mitochondria or not enough mitochondria. And so part of my research is trying to understand how this shape also allows them to do their function and produce the energy they need so that the neuron is able to do its function. And that's something that we still don't have a full appreciation for, but we know it's a phenomenon that mitochondria are constantly fusing together and going apart. And then you also bring up another point is when does this happen and where does this happen? So most mitochondria are actually originated in the soma. And so the soma is the part of the cell that has the nucleus. And so the nucleus is where all the DNA is stored. And a lot of the proteins that mitochondria need are found within the soma or by the nucleus. And so those proteins are translated, so they're made in the soma, and then they're moved about to the different parts of the cell. But we still don't know, how does that work when you have mitochondria that are really, really far away from the cell? How do they stay healthy when all of the protein and all of the DNA they need to make new proteins is all the way in the cell body, or the soma, which can be meters away in the human body? For example, you can have neurons all the way down at your toes, and they can be connected all the way up to your brain. But we still don't know, how is that connection, which can be meters long, maintained when you have these mitochondria that are super, super far away, but yet they need to be healthy. And so that's part of my research, is trying to understand how mitochondria can be healthy even when they're really, really far away from where they originated. That was a really great question, Brianna. Are there any other questions for me on where mitochondria come from or how they're distributed? All right, cool. We'll keep going, but feel free to ask more questions. So that was a really great segue into how do neurons distribute mitochondria. And so here I have a picture of a neuron, and we have right here different dendrites. And so these dendrites, you can see they kind of branch out, and it looks like a tree which has branches. So these branches are going out into different areas. And you can see here on this video on the left, which is a video of a dendrite. And we have the dendrite shown here in dark, and mitochondria shown here in white. That we have mitochondria moving into and out of this dendrite segment. And so before I get into all the details about how I got this image, you first need to understand where I got it from. And so I study mitochondria using Drosophila melanogaster. Drosophila melanogaster are known as the fruit fly. And the fruit fly is a really great model organism. So a model organism is an organism that scientists use to study different phenomena. Fruit flies are a really great model organism because they're really small, and you can get millions of fruit flies in a very small space and very cheaply because they live off of bananas and cornmeal. So I study fruit flies at Columbia, and Columbia has a long history of studying fruit flies. So fruit flies were first started to be discovered, or first used at Columbia in the Thomas Hunt Morgan lab. So he studied fruit flies in the early 1900s, and he later won a Nobel Prize in 1933 for his work on Drosophila, or fruit flies. The Nobel Prize is a prize in science that is awarded annually to scientists who make discoveries that impact the world around us. And so his discovery was looking at inheritance. So inheritance is when we have genes, which are the encoding of different proteins or DNA, the encoding of different proteins that we need on chromosomes. And so Thomas Hunt Morgan identified in fruit flies that the white gene, which is the gene that encodes the pigment in the eye of the fruit fly so that it's white or red, is actually on the same chromosome that tells the fly whether it's a male or a female. So how does this work? So he looked at white-eyed fruit fly females, which have 2 X chromosomes. And so on each of these chromosomes, this female fly had a gene that encoded white eyes. And Thomas Hunt Morgan crossed this fly, or mated it to a male that has red eyes. And males only have 1 chromosome of the X, and they carry 1 chromosome of the Y. And so for all of the females that resulted from this cross, we have 1 copy of X that is the white gene, and 1 copy that has the red eyes, or the red gene. So all of these female offspring had red eyes because the white gene, the red actually is more dominant than the white. And so if you have 1 copy of red, your eyes are going to look red. Whereas all of the males that resulted from this cross had white eyes. And they had white eyes because they only have 1 copy of the X, which they got from their mom, which has white eyes, and they got the copy of the Y from their dad, and that doesn't have any sort of red or white eyes on it because the red or white gene is only on the X chromosome. So Thomas Hunt Morgan was able to figure this out in the early 1900s, that certain genes are located on chromosomes, and these chromosomes can be linked so that certain genes are inherited with other genes. And so in this case, he found out that the white gene is also inherited on the sex chromosome, the X chromosome in Drosophila. So this is a good just sort of background to show you that fruit flies are a great model system because we know quite a lot about their genetics, but also that they've been well studied at Columbia in particular here in New York City. And so that's a really great fun history that comes along with this model system. So how do flies actually get stored? This is kind of a fun part of the lab. We have a little room, and this is where all of our fruit flies live. And so this is a space in the lab that's really high humidity. So we try and make it like a little tropical oasis for them. So we have a different heat lamp, and we have lights that go on for 12 hours so they get plenty of sleep. And they live in these little bottles, and at the bottom of the bottles, they have all the food they could need. And within those bottles, we have probably around 10 to 50 different Drosophila living in those bottles. And we have them stored in this special room so they can live in a very comfortable environment. And so they're treated really well. And so part of my research, I look at Drosophila under a microscope. And so we take them out of their bottles where they're living, and we put them under this microscope. And using this microscope, we can actually look at them and see different traits about them. So a microscope is a different technique that we use to visualize a specimen. So fruit flies are really, really tiny. They're less than half the size of your fingernail. And so if you want to see them, you're not just going to be able to pick them up and look at them. You're going to have to look at them underneath a microscope. And so within a microscope, we have these various parts. So we have a light source, and this is a light that beams up onto the sample. And then we have a base, which holds everything together. And we have focus, so we can move the specimen up and down relative to an objective. So an objective is just a series of different mirrors. And so these mirrors allow us to tune the angle of the light that is reflected from the sample. So we can move it into our eyes using this eyepiece here, and we can actually visualize the sample at a higher magnification. So that means we can basically zoom in on the sample and we can see it, and it can be much bigger than it is with our naked eye. So a microscope is really important in a fruit fly lab because when you have all of these different flies and they have different characteristics like red eyes or white eyes, you need to be able to distinguish between them. And so we use the microscope to help us sort out the flies and to help us do our daily work. A microscope is really important to a lot of different labs, but it's really critical for a fruit fly lab to have a couple of these, just so we can actually visualize the flies themselves.
Can I pop in another question?
Yeah, go for it.
How do you keep them from flying away?
That's a really good question. So actually in this picture here, you can see I have a setup, and this is called a fly pad. And so we anesthetize the flies with CO2. This is a harmless sort of process. It doesn't harm the flies. It just sort of knocks them out and it makes them go a little sleepy. But it doesn't kill the fly. And so what you can see here is we have this pad, and it sort of distributes the gas CO2 onto a surface. And then we tap the flies onto that surface, and they just kind of lay there. It doesn't really hurt them, and it keeps them from flying around all in the lab. I will say, though, that in any fruit fly lab, you will always have a few spare flies flying around because it is impossible to always capture all of the flies. We work with probably millions of flies, and so we probably have a few here and there that get out a little bit. but they don't survive long outside of their, comfortable environment because we don't have food laying around in the lab. So it's usually not too big of a problem, but you do have to be aware of making sure you're careful with putting the lids on them and closing the door to their comfortable habitat.
Awesome, thanks.
So once we use this microscope, we're able to sort out our males and our females. We're able to sort out the flies that have red eyes or white eyes. And we can also use this, a different type of microscope, to actually look at the mitochondria themselves. And so this is a very fancy microscope, but basically it's a version of the microscope that I showed on the previous slide. This one just has a lot more mirrors, so it's a little bit more expensive, and it also has some lasers. So here's a laser light source. And the lasers help us because we can actually visualize the mitochondria themselves, because mitochondria are really small. They're microns small. And so within a cell, we need to be able to visualize individual mitochondria. So we can't just use that microscope that I showed on the previous slide, because that would only show us about the size of a whole fruit fly, or maybe about the size of the head of the fruit fly. You can't actually visualize the mitochondria using that microscope. So using this setup here, we have an eyepiece on this side, and this is where I would look down into the sample. We have the different compound objectives. And so it's the same sort of setup here where there's a series of mirrors. So we have different sets of objectives to look at different sizes of samples. And then we have the sample here on the sample holder, and we actually move this over, we put the fly underneath, and we're able to adjust where the sample is underneath the objective. We have lots of different wires and cords going to and from this sample. And we also have a sample stabilizer. This is this really cool table that helps us to keep everything stable. And so it's really important when you're looking at really small mitochondria that it's not bouncing around, right? Because we want to keep our sample really stable. And so in order to do that, and so to keep everything stable, we have to keep it on this table. And this table has pressurized air going into it, and it keeps everything flat. So there's natural vibrations, everything from the subway going by down the street to the elevator going up and down. All of those things in the building that could disrupt the stability are sort of minimized using this setup. So how do I actually watch mitochondria move? Because if I have this microscope set up, that's really great, but how do I actually see the mitochondria? And so I use a protein called green fluorescent protein, or GFP. GFP is this special protein that when it makes a specific conformation or a specific shape, emits this really great green color. And here you can see GFP used in all sorts of model systems. So we have it here in Drosophila fruit fly. We have it here in a worm, C. elegans, in mice, zebrafish, and all sorts of things like a plant and different cells. And so what you can see here is in these images, they're staining most of the organism with this GFP. But what I do is I look at just staining the mitochondria with the GFP. And so that means that I have mitochondria that are shown with this bright green color, the GFP. And then I have other parts of the cell, like the cytosol, or the part of the cell that the mitochondria are in a different color. So I show the cytosol here in magenta. What we can see is we have mitochondria with this bright green GFP moving through this brightly colored pink magenta neuron. And you can see we have mitochondria that are moving. This is around 100 times speed, so they move a bit slower in real time. But you can see there's mitochondria moving in both directions of the cell, and they're zooming about in different areas. So this is how I watch mitochondria move, and I measure it using that microscope that I showed previously with this GFP tag on the mitochondria. So one of the main questions in my research is to look at this movement of mitochondria. So I showed you the movement of mitochondria in one cell type, but there are so many different cell types within the brain. And so one of my questions is, how do mitochondria move in these different cells? So we have cells that have different shapes. For example, these cells are really quite branched on the edges, but have these long processes which connect to each other. Other cells are a different shape, and they have a smaller subtree branch, and they have this other process. They have another subtree kind of branch, and then they have a different process. And so we want to understand, how do mitochondria know where to go in these different cells that have all these different shapes? We also want to know, how do mitochondria move across different age cells? So our older cells, do they have more mitochondria moving through them? Do they have less mitochondria? Are they moving faster or slower? These are all questions that we still don't quite understand, but using the fruit fly, we can get a better appreciation for how mitochondria are distributed and why they're moving to different areas, and how that's all controlled within the cell. And so using that in vivo imaging, or in vivo meaning that it's in the actual organism itself, I can actually see how mitochondria function in a normal cell. And that gives us a better understanding of not only how mitochondria function in a healthy cell, but how we might be able to get treatments for when there's an issue in the cells, like there's a disease in the brain, and how mitochondria might contribute to those diseases and illnesses. So I have a really fantastic team that I enjoy working with. So I have my PI, who is a principal investigator, Dr. Aaron Barnhart. And then I have my undergrads. So these are students who work in biology, who help out in my research, and they provide a really integral part to what I do every day. And their names are Nicole, Jordan, and Nermeen. So Nicole and Jordan both want to become doctors one day, and they think being in the lab is a really great way for them to explore working in sort of a medical system. And so they're really interested in the clinical applications for my work. Nermeen is a first-year student at Barnard, and she's not quite sure what she wants to do, but she's really excited about being in the lab and getting to work hands-on with fruit flies, and she finds that really exciting. I have some other great team members, including Adam, who's another graduate student, Vicky, who's a tech, and Ike, who's an undergraduate who's interested in working on computers and machine learning. So this is a fantastic team, and I really think that without them, I wouldn't be able to do the work that I do. And you should know that science is a really collaborative process, and so I get to work with these people every day, and they help me to better learn and better communicate my work. So I'm forever grateful for that team. So if you have any other questions, I'm happy to answer them. I know it's a beautiful day outside, and I'm sure you want to hurry out, but I'm really happy that you're able to come and listen, and I really appreciate any questions you have.
And I have one question going like way back to, I think, your second slide.
Great.
Or something like that. You don't need to go back. It's but so before, you said that cells originally took in mitochondria, that used to be bacteria. Do you know, like, what did cells do before that to get energy? Because I'm assuming they still needed it, right? Or was that something like we evolved into needing, or do we not know?
That's a really great question. So this is still a theory, and so we still will probably never actually know how this actually happened. But right now, what we do know is that eukaryotic cells, or the cells that are found in you and me and many other animals, use aerobic respiration, and that's because they use oxygen. But what we think is that either these organisms used anaerobic respiration, which is they used other processes that don't involve oxygen, so they don't involve mitochondria, or they perhaps had a different mechanism that just wasn't as good as the mitochondria. And so what we think is they were probably using anaerobic, so they were using without oxygen. And so other pathways that don't use oxygen don't need mitochondria.
Gotcha.
But that's a really great question. And it's kind of a theory. We'll probably never really know what happened, but we can probably guess based on what we know about the current processes.
Cool. Yeah, I imagine it was so long ago, like you can only infer so much.
Yeah, so it's probably around, I believe it's like 1.5 billion years ago. And so obviously we won't have traces of those original sort of cells that took in these bacteria. So we'll probably never have evidence for or against whatever theory scientists come up with. But we can base it off of what we know about current bacteria and current cells. And there are some cells that don't have mitochondria that use other processes for aerobic respiration. So it's not the only way that cells are able to function. For example, red blood cells don't have any mitochondria. So there are a lot of cells that don't have mitochondria and can still function just fine. They just use other processes to have energy.
Gotcha. I think we also have another question in the chat. Lucas is asking, doesn't keeping the Drosophila inside a small place interfere with their metabolism, stress, or epigenetics?
That's a really great question. So like Lucas said, we do keep the mitochondria in a small place. So here's a picture of how we store them. You actually can't see in this picture. You may be able to. The fruit flies themselves are really, really tiny. So they're just the little brown specks here. And so that's a really great question. So we have these lab model systems, and we do the best to keep them in a comfortable environment so they're not stressed. So we keep them on a 12-hour light-dark cycle, which means they get 12 hours of light and 12 hours of dark so they can sleep comfortably. And then we also give them all the food they need, and we give them a proper nutrition. So it's a balance of cornmeal and bananas and other different fruits and vegetables. So they get their balanced nutrition. But they can still have stress, right? So being in a lab isn't always the most comfortable, and they're not in their native environment. But they also don't have predators, so they don't have to worry about finding food. They kind of lead a posh lifestyle. And these flies probably wouldn't survive well out in the wild because they've been bred in captivity for so long. But Lucas's question about epigenetics and how stress may impact our research is really important. There's a couple other labs at Columbia's campus that actually study stress in mitochondria, and specifically not stress in mitochondria, stress in Drosophila and sleep, which is actually really interesting. So we try and keep to the best practices of what we know that fruit flies like, and we hope that this recapitulates their natural environment. But that's still something that's going to be discovered, and we're going to have to look at how model systems actually maybe not completely recapitulating the natural environment because they do have differences in their genes because they've been bred in captivity, and differences in stress because they're not in their native environments. So they have different stresses than maybe their native counterpart would. So that's sort of a roundabout way to say, Lucas, we're still trying to figure everything out. We do the best we can to keep their stress low and to keep them comfortable, but we probably won't know the full extent of those differences until we're able to do further studies. And some labs actually do capture fruit flies from the wild and look at them. And that's not something my lab does, but it's definitely something will be interesting in the future to compare our studies with others that compare them to wild type fruit flies.
Does anyone else have any other questions?
In the... Oh, do you isolate their mitochondria and how? Yes, that's a really great question. So in the images you see, I'm looking at individual mitochondria because I'm tagging them with GFP. But another part of my research is actually going to be isolating mitochondria and looking at their bioenergetics. So looking at the different byproducts that are used for ATP synthesis. So looking at how productive mitochondria are in different environments. So looking at mitochondria in young flies and old flies. And actually, we can use the same technique to isolate mitochondria that we do to visualize them. So we visualize them using this green fluorescent protein, or GFP. So we tag the mitochondrial outer membrane protein, which is a protein that's found on the surface of the mitochondria with GFP. And we can actually use this tag to purify mitochondria and look at how their bioenergetic function is. So we can see how productive mitochondria are in young flies versus old flies. And part of my research is actually going to be looking at isolating mitochondria using this GFP tag in axons and dendrites. And so I'm going to find a way to develop a tag that's specific to specific parts of the cell, either the axons or the dendrites. And then I'm going to look at the function of those mitochondria based on that tag. So are the mitochondria functioning better in the axons or the dendrites? And how does that function differ? So that's a really great question, Lucas. And that's something that I'm really interested in and will be working on throughout my research here at Columbia.
That's really cool. Just to follow up, is that like an antibody sort of like where you're pulling away the mitochondria, or like a centrifugation where you're spinning them and like taking a layer of mitochondria out?
Yeah, so it's actually both. So what we do is we get the fruit fly, we actually mash up its head, so it does have to die in the process, unfortunately. And then we do a process of steps where we wash it to get away all the other bits and proteins we don't want. And then we do centrifugation. And so centrifugation is where we spin the sample really, really fast. And the dense parts of the sample are going to go down to the bottom. And we're going to look at the layer where we think the mitochondria are. And we're going to put that layer through, and we have an antibody. So an antibody is a specific protein that's going to attach to the GFP. And so it's not going to attach to any other proteins. It's only going to attach to the GFP. So once it attaches to the GFP, we're going to wash all the other proteins away. And that way we'll be able to select for just the mitochondria that are tagged. And so we use a combination of steps of washing, centrifuging, and then using an antibody. And so that antibody is able to help us separate all the other proteins and maybe the untagged mitochondria from our tagged mitochondria.
Very cool.
I see there's another question from Lucas as well. You said you label mitochondria with GFP, right? Can you label xenobiotics or substances too to assess their impact on bioenergetics, and how? That's a really great question. So yes, we definitely label mitochondria with GFP, but there are other tags that we can use to label mitochondria and other substances. And so one of them is doing C13 labeling, so labeling with a radioactive carbon. And so we can put this radioactive carbon into their food, and we can look at how that radioactive carbon is input into their whole body over time throughout development. Or we can do other labeling, such as labeling with what's called an HA tag or a flag tag. These are basically different amino acids or different sequences that have a very distinct structure. And so it's like an antibody where we'll have a very specific sequence that matches with a very specific tag. We're able to purify our mitochondria using those different techniques. So we could do carbon labeling with radioactive carbons. We can look at GFP expression using a mito-GFP antibody, or we can look at various tags like an HA tag or a flag tag. So all these techniques are probably going to have to be optimized. They've been done in some other studies on fruit flies, but not necessarily for the purification of mitochondria. And so that's where a lot of my research is going to be optimizing these techniques and trying to figure out how I can specifically get mitochondria from axons and dendrites.
Anyone else have any other questions?
I have a question. So, Avin, so you mentioned before, like, something that blew my mind that I didn't realize was that mitochondria are relying heavily on proteins made from the genome of the cell that it's inhabiting. So
Yes.
that's really interesting because you can imagine that the proteins a neuron makes are very different from the proteins a kidney cell makes. Yet both cells are making the proteins, I assume, necessary for mitochondria to survive.
Yep.
So has anyone looked at, like, is that true? Are different cell types, for example, like a kidney cell compared to a neuron, making the same exact proteins that a mitochondria needs to survive? And when you say that you look at what happens to mitochondria when a cell ages, I guess it's in respect to its host cell aging, right? So I guess that's what I'm imagining. Like it's aging and maybe whatever proteins it's needing from its host cell are just maybe not being made, or I don't know. Yeah.
Yeah, those are great questions. So different cells, they all have the same DNA that is used to make mitochondria. So mitochondria actually are really cool because they have their own mitochondrial genome. And this we think is related to how we had the bacteria originally as sort of the origin of mitochondria. So they probably had their own genome. So much of that is degraded / it's gone to the nuclear genome. So most of the proteins that mitochondria need reside in that nuclear genome, which is encoded in the cell's nucleus. But some of the proteins that mitochondria need, actually 13 of them, are encoded in a very, very small mitochondrial genome. But all cells have that genome for encoding the necessary mitochondrial proteins in their nucleus. And all the mitochondria have a small part of their genome encoded in their mitochondrial genome. So they all types of cells have the necessary ingredients to make all the proteins that the mitochondria needs. But what's likely different between these cells is the levels of those proteins. So the expression levels of those proteins can differ significantly. For example, the surface proteins, which allow the input of different ingredients like glucose, may be different in the brain and they may be different in the liver. And so that could allow various levels of glucose or various levels of oxygen to enter the cell. And so this is where you see some differences in the mitochondria and different parts of tissues because they have different expression levels of these proteins, which allow for different functions. And so looking at those expression levels or looking at the different amounts of proteins in the mitochondria and different cell types, we can kind of understand how these mitochondria may function differently in different parts of the cell or tissue. And then the second part of your question, Sergio, was about having differences in aging mitochondria and aging cells themselves. And so this is really interesting because mitochondria are, they live much shorter lives than the cell itself. And so like Abriana mentioned earlier, we have mitochondria that fuse together, they come together and they go apart. And when they fuse together, those proteins mix. And so it's kind of like if you have a car and you start replacing all the car parts, like you start replacing the tire, you start replacing the wheel, you start replacing the cushions. Like after a while, it might kind of be like a new car because you have all these new little parts that are added to it. And so if you add enough new parts to it, that mitochondrion is sort of part of the old mitochondrion, but part of a new one. And that's what we think happens to a lot of these mitochondria. So in order to stay healthy, they kind of mishmash together and sort of the old parts get reused and the new parts kind of help to aid in that process of recycling. And so as that cell or tissue ages, the mitochondria within it are going to meld together. The new tissues or the new proteins are going to come together and the old ones are going to go apart. But as the cell ages, those proteins and that sort of cycle of renewal sort of may change in different parts of the cell. And that's the part that's really interesting because there's been some studies that have shown in vivo that mitochondrial motility actually decreases with age. And so that's really interesting because what we don't know is if motility is decreasing, then that means that there's fewer mitochondria that are moving through that are healthy with those fresh new proteins. So how are mitochondria staying healthy if there's fewer fresh proteins being delivered? And so that's part of the question, sort of ongoing, interesting part of the field is how do we have these mitochondria that can, sustain the life of the neuron, which has to survive the lifetime of the organism when we see decreases in the motility. And so that's a really great question. There is sort of a relationship between the aging of the cell and the aging of the mitochondria, but they're also kind of different because the cell stays, has to stay alive the whole time, whereas the mitochondrions can mishmash together and have little parts renewed, whereas the cell has to stay intact and the same throughout the entire lifetime of the organism.
Wow, thanks, Alvin. Sounds like they're not just the powerhouse of the cell.
Yes, exactly. Yeah, you hear the phrase of mitochondria, the powerhouse of the cell, but they actually do so many different things beyond just producing ATP. They do all sorts of things to help maintain our cells and keep them healthy. Well, thank you all so much for those questions. Those were really great questions, and I think they helped us to all understand how mitochondria work in neurons.
Thank you so much, Alvin. That was very cool.
Woo. Thank you,
Cool.
everyone.
Okay.
Thanks,
Thank you
Alvin.
everyone for coming. If you have more questions, feel free to ask it. If not, feel free to log off and we'll see you at our next event. Cool.
Have a great weekend, everyone.
Thanks for
Bye,
coming,
everyone.
everyone.